Hey everyone, I'm Julian, your Biology 2600 lab instructor. And in this video, I'm going to cover the material for Lab 8, Nutrient Cycling. So last week we looked at food chains, specifically how organisms at higher levels in the food chain affect organisms at lower levels of food chain. This week we're going to continue on from that, but we're going to focus on the transfer of energy within those food chains. So the purpose of this lab is to highlight the connection between plants that exist on a riverbank and the river. So we're going to start by determining the quality of plant letterfall that enters the river. And we're going to do this specifically by looking at the carbon-nitrogen ratio. And then we're going to look at how the quality of the letterfall affects its decomposition rate and what role invertebrates have in this. So before you watch the rest of this video, please read the Lab 8 file and the appropriate sections in the textbook, and also refer to the appropriate lectures from Dr. LaRue. So in Lab 7, I showed a very simplified and generalized version of a food chain, one which highlights the different categories. So primary producers, primary consumers, secondary consumers, and in which the arrows point to the food source. So predators eat herbivores, herbivores eat plants. Today we're going to take a different spin on this, and we're going to look at the transfer of energy. So the sun provides all of the energy for primary producers, and this includes plants, algae, cyanobacteria, etc. And by undergoing photosynthesis, these organisms produce sugars, which, when broken down, release energy. So when primary consumers like herbivores eat plants, they obtain some energy, and when secondary consumers like carnivores or predators consume herbivores, they obtain some energy. Now, a really interesting concept is that very little of the energy that is produced by plants is actually passed along to primary consumers, or in this case, herbivores. So 10% of vegetation is actually eaten by herbivores. The remainder, so 90%, actually ends up falling to the ground as dead or dying litterfall, as is depicted by this lovely picture here. So you can see the ground here is covered in dead or dying litterfall. Of course, if trees happen to be close to a river, some of that litterfall will actually make its way into the river, as you can see here. Once in the river, these dead or dying, leaves end up being a food source for invertebrates, specifically shredders, as we discussed in Lab 2. So the first part of the experimental procedure for this lab is actually going to be collecting this letterfall so that we can assess it. And this is simply done by placing a basket under some trees next to the river. Of course, I'm going to go through that in a bit more detail in just a little bit. So as I briefly mentioned, some of this letterfall makes its way to the river and provides a food source for invertebrates. But just like every one of you, different types of leaves are special and unique in their own way. And in this case, some of them are higher quality than others. So the way we assess quality is by looking at the carbon to nitrogen ratio. So the proportion of carbon in relation to the amount of nitrogen. So leaves like alder, for example, are considered higher quality because they have a low carbon-nitrogen ratio. So what that means is there's more nitrogen relative to the amount of carbon. And this is important because carbon can be pretty much found everywhere. It's not a limiting resource, so it's easy to get carbon. Nitrogen, on the other hand, is a limiting resource. So the more nitrogen you get, the higher the the leaf or the food in general is. So alder is high quality because of a low carbon nitrogen ratio. Chocolate pear leaves, on the other hand, are considered low quality because they have a high carbon to nitrogen ratio. So again, meaning that there's a lot of carbon in proportion to the amount of nitrogen within these leaves. So I'm going to go through this in a fair bit of detail in a little bit. But essentially, we took different types of leaves, put them in bags, and we submerged them in the river for a period of time. So, as mentioned, these leaves are a food source for different types of inverts, specifically shredders, even more specifically, something called detritivores. So these are organisms that eat dead or decaying plant or animal parts. And by doing so, they convert coarse particulate organic matter to fine particulate organic matter. So that's just a complicated way of saying they break it down into smaller pieces. So as you can imagine, if you have a higher quality leaf, you're going to end up attracting several more inverts than you would for lower quality leaves. And if you have more inverts eating these leaves and converting them to smaller bits, you're going to end up getting higher decomposition rate. And so this is the second part of our experimental procedure. And so just to kind of finish this off and tie everything together, the quality, if you get a higher quality of leaves, so if you have trees that produce high quality leaves and they fall in the river as letterfall, they're going to attract more inverts because they're a better food source. And if you have more inverts, you're going to attract more secondary consumers like fish. And so just by having better leaves, you're going to end up affecting the entire system from the bottom up. And so, of course, all of this nutrients gets recycled. So the inverts break C-palm down in F-palm, and then the F-palm is actually broken down into the raw minerals like carbon, nitrogen, and phosphorus by bacteria and fungi, which are decomposers. And all of these minerals go back into the soil, which is utilized by plants, and the whole nutrient cycle starts over again. And so for your convenience, to help reduce confusion, I made this pretty little figure, which essentially summarizes both of the experiments. So part one's on this side. These are the main steps. Part two is on this side. These are the main steps. So now I'm going to show you where the experiments were done, and then videos of me actually doing the experiments. So here is the majority of Memorial University's St. John's campus. You got your science building, Newport Science Facility, and we're going to go to Portugal Cove St. Phillips, specifically Healy's Pond Crescent. So this is Healy's Pond, this is Healy's Pond Crescent, and this is the little stream that we're going to be doing most of our experiments. So I'll see you there. Hi everyone, here we are at Healy's Pond Crescent. The river is behind me. So we are here to do part one, the litter fall in the litter basket. So we're going to put a basket next to the river, collect some litter fall, and see what we get. So this is your litter basket, nothing special, it's metal so that it's durable. It has holes in it so that rainwater or pond water, whatever, doesn't accumulate. It has little holes on the ends so that you can tie a string onto it so that it stays in place. We have our property of Memorial University so that nobody takes it and tries to do our experiment for us or anything like that. So that's essentially it. I'm going to take this basket walk over to the spot next to the stream. We'll start a video there, and I'll see you there. Alright, so here we are next to the stream. So see the stream there? We're essentially right on the edge. There's no specific criteria for where you place it. There's going to be different trees everywhere, so depending on where you place it, you're going to get different types of litter fall, which is part of the fun. So you want to keep that in mind. The other thing is, and probably more important, is you don't want to have it too close to the water, because even though it's not in there now, if there's a heavy rainfall or something like that, the basket could get submerged, or even worse, swept away, or the leaves could get swept away, and then you don't have anything collected, and then you don't have an experiment. So I'm just gonna use these strings, which are already on there. Gonna tie them to trees on either side. Gonna leave it there, and we're gonna come back in a few weeks to collect it, and we'll see what we get. Super exciting! Hey everyone, here we are back at Healy's Pond Crescent to collect our litter basket. So significantly colder than before. You should kind of be happy that you're not out here. So I'm gonna go grab it and we'll see what we got. Alright, here we are at our basket. So we did end up getting a fair amount of stuff. Different types of leaves, which is good, and of course it really depends on where you place the basket, what trees are next to it, etc. How far away from the water, time of year, all that sort of stuff. So I'm gonna cut the strings, gonna grab the basket, and we are gonna head back to the lab. So make sure that you, if you are doing this, that you take everything with you that you brought out here. Save the environment, all that stuff. So, got all that. Alright, I'm gonna walk back and I will see you guys at the lab. Hey everyone, here we are back at the lab at the litter basket. So this litter basket has been sitting in the front of the lab for 1 week, and now we're going to empty it out. And we're going to spread it out. So this is actually the most tedious part. So you guys are complaining about iron, you should be happy you're not doing this. So what we're going to do is separate the leaves out by type, and then we're going to measure how much they weigh to get an idea, and then we're going to combine all of those things to get an idea of how nutritious this litter fall is. So I'm not actually going to do that. I will show you the ID guide that you will use in just a second, and then I will talk to you about how to actually do your calculations with some sample data. Alright. Hey everyone, so I did sort of just pick out a few leaves in this little pile. So these all look like birch leaves to me. So you can imagine, for example, if you have leaves like this, that's pretty broken down and it's kind of hard to tell exactly what it is. And then things like this, if you try to peel those apart, they're going to break up and it's going to be pretty hard to identify them. not that it isn't hard to identify leaves anyway, but so you can see that there's going to be a decent amount of error in this process. So we use this guide right here to do it. These are commonly found leaves of commonly found organisms that occur in the riparian zone. So essentially you pick out unique features of each one, and you try to identify the leaves or fit them to one of these categories. So, for example, birch leaves are, as I pointed out earlier, they're wide at the base and then they go off to a tapered end, whereas these are not that wide at the base, they're kind of wide at the middle. You have spruce needles, which are squarish, and they're going to be smaller than fir needles, which are flat. Pine needles look essentially like eyelashes, they're super thin. Sweet gale has a unique end. So things like this, and then you kind of look for them and you figure out what's what. So once you have all of your leaves, all of the leaves of a certain species picked out, you make them into a pile and you weigh how much you weigh them, and then we're going to use values from already calculated values to figure out their carbon and nitrogen ratio, and then we're going to combine all of the different types of leaves and the carbon nitrogen ratios and weights of all the different types of leaves to get an idea of how nutritious this entire litterfall sample is. All right, so I brought up the lab file here. I'm just going to show you the data for this experiment. So if you scroll all the way down to the bottom, you're going to go to you're going to see an example table, and then you're going to see table 2. So the example table just shows you how to get each of your values. So as mentioned, the carbon nitrogen ratios have already been calculated for all of the species that we're looking at by MUN's Create-It lab. So we already have those values, and those are going to be given to you. And you're also going to be given the total mass of each type of leaf. So this is an example table because the species here are just made up. They don't refer to anything. The purpose of this table is just to show you how to do the calculations. So the actual table that you're going to fill in is down here. So as mentioned, you're given the carbon nitrogen ratios, and you're also given the total mass of each specific type of leaf. And these are, in fact, real carbon nitrogen ratios for these specific species. So this was for the basket that I did, and I just made up a couple of other baskets just so we have something to compare it to. And I'll talk more about that in the discussion. So these calculations down here, highlighted in blue, show you exactly what to do. So if you have species 1, you have a carbon nitrogen ratio of 36:1. That means for every 36 carbon atoms, you have 1 nitrogen atom in the leaves from this specific species. And so just for fun, we picked a total mass of 10. So to get your grams of nitrogen, you go your nitrogen value, which is 1, divided by the carbon plus the nitrogen value, so 36 plus 1, which is 37. So 1 divided by 37, and then you multiply that by 10 because we have 10 grams, and that gives you 0.27 grams. So based on this ratio and this amount of leaves, we have 0.27 grams of nitrogen in our leaves. And then to get the grams of carbon, it's much easier. You just go 10 grams, because that's your total, minus your grams of nitrogen equals your grams of carbon. So we're only looking at 2 ratios. So if this is what you have for nitrogen, and this is your total, then it's pretty easy to get your grams carbon. To get your total N, total mass of N in the bucket, all you do is just add up all your grams of nitrogen. To get your total C, all you do is just add up your individual grams of C, and you get that. And then to get your final overall ratio of your carbon nitrogen ratio for your whole basket, you just divide this by this. So you're essentially normalizing it to N, which will give you a value of 1 for N, and then this is your value for carbon. So you guys are going to perform these calculations for your basket. So work out your grams of nitrogen for each of these, work out your grams of carbon, and then you're going to get an overall value. And for your discussion, you're going to compare it to these two. And I'll talk more about that in a second. So that was part 1. Now we're going to go through part 2, starting with the location. So again, Memorial University, St. John's campus, and we're actually going to collect our leaves just off Bennett Road. So if you remember Bennett Road, that's where we did Lab 2. So this was the stream for Lab 2, and this was, sorry, the riffle and the pool, and the little bridge, and where I did my talk over here. This time we collected leaves over in this area, and this was just picked at random just because it had 3 of the different types of leaves that we needed. So once the leaves were collected and processed, then we went back to Healy's Pond Crescent, just like in the first part, and they were submerged in the stream approximately right here. Hey everyone, I am here just off Bennett Road. So this is across the street from where we did Lab 2. So I am here to do part 2. Whoa, it's windy, and of course it just started to rain. But anyways, here to start part 2 of Lab 8, which is the decomposition for letterfall. So the first step here is to collect the leaves. So we're going to collect some birch leaves. I am, and I know you're jealous because it's super fun. Birch leaves, chocolate pear leaves, and as you can see behind me, alder leaves. So just take a plastic bag and you just pick leaves essentially one by one until you think you have enough leaves to put in the decomposition litter bags. So I'm not gonna show you me picking leaves, but trust me, I did it and I had super time. I will show you what the leaves look like next, and then next time you see me, we'll be back in the lab when we're measuring them out. Alright. Alright, here we are back a week later. So the leaves have been, the leaves that were collected have been sitting here for a week. So they're dried out. You can hear they're kind of crunchy. So we're going to use a scale. And you essentially are going to add enough leaves so that you have 5 grams. So we added, I mentioned that I need to collect 10 grams, and that's because we have 2 different bags to fill. So, and each bag has 5 grams. And so you can get exactly 5 grams or close to. If you can't exactly get exactly 5 grams, you could just set your initial value to whatever the weight was. we're gonna pretend I got exactly 5 grams. So you take your big bag and you put all of those in there. So that's that bag ready. And then we're gonna do the exact same thing. We're not gonna do it because you know the idea. So you get another 5 grams. We're gonna put it in the smaller bag. Then we're gonna put, then we're gonna do it for the birch and the chocolate pear. This is the alder here. So we're gonna have 2 bags each, 3 types of leaves, so 6 bags in total. We're gonna put them on a rope, and then I'm gonna go put them out in the river. Hi everyone, here we are at Healy's Pond Crescent. This is the same location that we set up our the first part of our lab, the litter fall in litter baskets. Today I am setting up the decomposition in litter bags, part 2. So I already have everything set up here. So we have 3 smaller bags. They have your chocolate pear, birch, and alder. And we have 3 larger bags, which also have those 3 same species. And so what I mean by smaller and larger, of course, is these have 2mm holes, whereas these have 1mm holes. And we'll talk more about that later. So leaves are in bags, they're already on a string. So I'm just gonna head to the site and put them in the river. All right, here we are in the river. Got my bags, got a pocket knife. I'm gonna go over to one side of the river, tie it onto a tree which is stable-ish. Then I'm gonna make sure I have enough rope so that the bags are submerged, and tie it on the tree over on this side, which is also stable-ish. So, if I fall, you gotta promise not to laugh. So, as you saw, not as easy as it actually sounds. The string keeps wrapping up, the current keeps pulling the bags, there's branches everywhere, the rocks are unbelievably slippery. But last year, or a few years ago when I did this and I fell in, or the same year when I actually had to chase a bag down the river, it was much better than that. So, overall, a success. So we're going to leave the bags for a number of weeks, and then we're going to come back and collect them, bring them back to the lab, and calculate the decomposition rates. Alright. Hey everyone, here we are back at Healy's Pond Crescent. This is the last part of part 2. The decomposition in litter bags. So I'm gonna head on over to the site and collect the leaves that were in litter bags. Just lost my camera. So hopefully they're there, and I'll see you there. Here we are at the river where we tied our litter bags. So if you can see in the background there behind me, they're still there. One of my greatest fears in life, that I'll show up and I'll be gone. But they're still here, so I'm gonna go out and get them. Nothing special you gotta do, just cut the strings, make sure you take everything that you brought out here, and that's it. So, I'm gonna do that now. Cross your fingers that I don't fall. Here we go. Alright, so here we are with the bags that have leaves in them. So these are alder leaves. We have our 1mm bag, 2mm bag, 2mm holes. So what we're going to do, first thing you do is just rinse off the outside, We're not going to actually go for the actions, but you're going to insert the outside. And then you're going to empty the leaves into different size sleeves. So this one goes on top, has bigger holes than this one. Pull them this way. You're going to empty your leaves out. So the purpose of washing the outside of the bags is to get anything that's any foreign matter that's not supposed to be there. So we just want to look at the leaves and don't want to weigh anything else. So they're here, you can rinse them off. That washes off any inverts, anything like that, little bits of sand, dirt, etc. And all of that stuff is caught down here, of course. So if you wanted to look at the inverts that were found, you could do that. So once they're washed off, we're going to put them in a pan. And spread them out. And then we're going to put this pan in the oven. Back here. So the oven's actually turned off right now, but I will turn it on. And it's not like your house oven where we're not going to cook them. It's just going to go up to 50 degrees so that they can dry out. So if you remember, the first thing we did after we collected the leaves was to let them dry out in the lab for a week, and then we measured them. So we're doing the same thing. We're going to let them dry out for a week and measure them that way. And so then we're comparing dry leaves to dry leaves or apples to apples. So gonna leave them in there for a week and then we'll be back. Hey everyone, here we are a week later. So the leaves were dried out in the oven for a week at 50 degrees. So now we're comparing dry leaves to dry leaves. So we're going to compare the initial weight of the dry leaves to the final weight of the dry leaves after they have been submerged in the river, decomposition has occurred, and then we can see what the decomposition rate is. So I'm just going to pour these out here. And so we're going to get our value, we're going to record that in our table. So now I'm going to go through how you fill out your tables, and we'll go from there. All right. So to do the calculations for part 2, I brought up the lab file. We're just going to go, we're going to go down to part 2 section. And so I'll show you in a second, but I've used this formula to actually do the calculations for you. So this is your decomposition rate, K. In order to calculate that, you take the initial mass. So our initial mass was 5 grams for each of the leaves, divided by your final mass. And then you take the natural log of that and you divide it by time. So you're not going to have to do this for your table, but you will have to do this for one part of your discussion. So I'll point it out here now. It's important to remember that T is actually a value that is in years. So it just needs to be the year value in order for this equation to work out. So because we've been dealing with weeks, specifically 6 weeks, that's how long they were in the river, you're going to need to convert that to years. So 6 weeks divided by 52 weeks for a year equals 0.11 years. So keep that in mind, and I'll mention that again when I'm talking about the discussion. So now we're going to scroll down to table 1. So like as I mentioned, your decomposition rates have already been calculated, and they're listed down here. for each of your free species. Carbon nitrogen ratios are also listed here, and you have the size of your holes in your bags, 1 millimeter or 2 millimeter. All right, that's it. All right, so the last part is your write-up. So I'm going to go for that. So I've got up the lab file here. I'm going to scroll down to the write-up. So I'm not going to give you a lot of detail for some of these sections because you've done it before. So title, you should know that it should be detailed at this point. For your introduction, give some background information on nutrient cycling. You could take that directly from this handout. And you're going to formulate two hypotheses that answer these problems. So for those of you that still don't know how to make a hypothesis, I'll mention it again. The problem is always going to be a question, and to make your hypothesis, you just convert that question into a statement. So, is decomposition faster, blah, blah, blah? Your hypothesis will simply go decomposition is or is not faster in blah, blah, blah. And that's your hypothesis. Problem two, how does carbon nitrogen ratio affect litter decomposition rate? All you have to do is say carbon nitrogen ratio increases as litter decomposition rate increases or decreases or doesn't change or whatever. So your methods just simply refer to the lab file, lab 8, nutrient cycling, fall 2020. For your results, I'm going to go into a bit more detail. So I'm just going to show two of these lab files side by side so I can talk about different things at the same time. So your data is essentially in two tables, table 1 and table 2 down here. So for your results, you're going to use table 1 up here to make two graphs. So your first graph is going to be your decomposition rates for your free species in the 1 and 2 millimeter bags. So all of the information is here. You might have to rearrange it in whatever program you're using to plot it, but all the information is here. So I'm not going to specify which graph type you should use because I want you to actually think about it, and try to think about which graph type will show the trend in the most clear way. So decomposition rates from 1 to 2 millimeter bags. Your second graph is going to be your decomposition rates as a function of your carbon nitrogen ratio. So for this one, you can pick either 1 millimeter bags and use those 3 values, or you can use the 2 millimeter bags and use those 3 values. Entirely up to you. I don't really care. The trend is going to be the same. You're going to have the same type of graph, or the graph is going to look the same, so you can pick. The only thing you might be confused about is how to make it, how to get your decomposition rate to be a function of the carbon nitrogen ratio. So because the ratio is something to 1, all you have to do is pretend the 1 is not there. So if you're using 1 millimeter, go 5.15 divided by 22.5. That'll give you a value. And then this number divided by 27.1, and then this number divided by 62.7. So you're going to get 3 values for each of your species, and then you're going to plot that, and it's that simple. So again, think about what type of graph you should use. Graphs should be computer generated. They're titled as Figure 1 and Figure 2, and the titles appear at the bottom for graphs as opposed to tables where they appear at the top. And of course, make sure that your titles are descriptive and your axes are labeled appropriately. So, if you wanted some extra points, you could get those points by generating your graphs in RStudio and submitting those along with our code. So that'll get you 2 bonus marks if you want to do. If you don't worry about it. As always, in the results, you're going to include a summary statement. So one for each of these graphs, just summarizing the general trend. And the last thing you're going to do is fill out table 2 and include that in your results. So I talked about that earlier. So the example table shows you exactly how to do these calculations. So you're going to do that for these down here, and you're going to end up getting a value down here that we can compare to these. I'll talk more about that in a second. So the last part is your discussion. So your first hypotheses is based on if there's a difference between 1 and 2 millimeter bags for each of the 3 species. So you're going to start by saying hypothesis is or is not supported. That'll get you 0.25 marks. Easy. And then you're going to explain it. So in terms of explaining it, the things you want to focus on are, so the whole purpose of the different size holes here is that they're going to limit the type of inverts that can actually make their way into the bag to get at the leaves that they eat and therefore break down. So keep in mind that the decomposition rate is strongly affected by how much or how many inverts are actually eating these leaves and how much they're breaking it down. So whether they're actually consuming that or breaking off little pieces that are floating away, that's going to reduce your weight. So if you have 2 millimeter holes, they're going to be bigger than these. So you're going to do 2 things by this. One, you're going to let in bigger inverts along with all of the smaller inverts that can fit in the 1 millimeter bags. But you're also going to let more inverts go in at the same time, or in general, because these holes are bigger. And that is the trend that you're going to see. So if you just quickly look at these numbers here, you're going to see that the 2 millimeter for each of these species is a bigger decomposition, is a bigger number than the 1 millimeter, and that means the decomposition rate is higher, which means that they were broken down faster, and that is an indication that more inverts were eating them or breaking them down. And so you can also refer to this lovely diagram, which summarizes that as well. So, well, no, actually that doesn't summarize it. Never mind, scratch that. But that's essentially it. So that is for your first hypothesis. For your second hypothesis, it talks about whether the carbon nitrogen ratio affects it. So that's the one that you're going to refer to your diagram. So in this case, you're going to pick either 1 or 2 millimeter bags, and you're going to focus on the ratio. So the trend that you see here is that the lower the ratio, that means the higher the quality of leaves. So this is the lowest, this is a bit higher, and this is the highest ratio. So this is the highest quality, a little bit lower quality, and the lowest quality. And you can see that the highest quality is going to give you the highest decomposition rate. So this is the highest quality food, so more inverts are going to be attracted to this, and that's why this is broken down faster, as indicated by the decomposition rate. And so that's what's illustrated here. So higher quality leaves alder attracts more inverts, which equals a higher decomposition rate. And so the other thing you want to do is if your hypothesis is not supported, that's not a big deal. Just say hypothesis not supported, say this is what I expected to happen, and just write what I just told you. And then just give a reason. So like there's any number of reasons why your hypothesis wasn't supported. And you just pick one of those. The other thing, and so you're going to do that for both hypotheses if they're not supported. The other thing is, for this one, you're actually going to find an article that supports your um, results. So something in the literature that actually backs up your hypotheses and your results. And so this should be pretty easy to find because it's widely accepted that decomposition rates are very affected by the carbon nitrogen ratios. So provide specific detail in what they actually did in the article and their findings that support exactly what you're saying. Paragraph 3, I'm going to come back to that in a second. Um, Paragraph 4 is referring to table 2. So, so here you're just going to compare your overall values for your three different baskets. So the one you calculated and the other two that were provided here. Um, so a couple of simple questions. Does the basket with the highest overall carbon nitrogen ratio, is that the same basket that gave the most total nitrogen? Does that have to be the case? So you can kind of think about this. It doesn't have to be the case. And the reason is these are ratios. So if you have a basket that has a lot more carbon, then it can have more nitrogen than another basket, but the ratio can still be smaller. And that's kind of hard to explain in a video, but if you just play with the numbers a bit, you should be able to figure it out. Last paragraph is suggest one potential problem for each of the experiments and how to address that problem. So let's just shoot up to the hypotheses. So in terms of bags, it could be any number of things. So maybe there was mud in the area that covered up some of the holes, so half the bag was covered up and it didn't let certain inverts in. It could be that there was another source of nitrogen next to the bag, and that's why you didn't get the obvious trend with your carbon nitrogen ratio and your decomposition rate. The obvious one that most people are going to pick is it should be done repeated in different areas for consistency. You can use that one. One thing I'll mention is that you should give different ones two different reasons. So if you say repeat this experiment several times, you need to come up with a different problem slash improvement for your other experiment, just so you guys think outside the box. And so for paragraph 3, you're just going to do some calculations. So one of the reasons I added this is just so I can see that you guys can do your calculations. The other reason is just want to play around with some software to see if it actually works. So let's start it up. So you're going to have calculation 1. See if that comes up. Yep. All right. So this one is pretty simple. You're just going to use your formula to and plug in your values. So K equals ln of MO divided by MT over T. And it's coming up. And you're just going to plug in your numbers. So equals 5 divided by 2.84. You're going to take the natural log of that. You're going to divide it by time. Keep in mind that your time is actually going to be in weeks. Sorry, it's going to be in years, so you need to convert your weeks to years. So it's going to be 0.11, as I already mentioned, and then you just work that out and you get your answer. So I'll give you a hint. There's a hint here. That answer should be this value right here. So you're going to need to show your workings in your appendix, but you know what your value is. So that's good. So your second one is going to be a little bit more complicated. So you're going to start with your same formula. But you're going to need to manipulate it a bit. So the reason it's complicated is because you're using a natural log. So the one thing you want to remember is that your natural log, if the value is not uh, specified, the base of the natural log is e. So first thing so you want to solve for mt here, which is your mass in 13 weeks. So first thing you're going to do is bring your t over. So kt equals ln of mo and t. And then you're going to take your natural log. So again, your base is e. So if you divide both sides by ln, and it's coming up, what you will have is your k to the t, oh, broken. Your k to the t will be to the base of e, and you would just have mo divided by mt. And coming up, yep. And then the rest is easy. You just bring your mt over, which is multiplied by e to the kt equals mo. Divide both sides by e to the kt. So mt equals mo divided by e to the kt. And so you have all of these values. E is just on your calculator, it's like 2.712. K, your constant is given, it is 4.82. T, so this is 13 weeks, so you need to go 13 divided by 52, and that'll get your value here. And your initial weight was 5 grams, and that'll get your final. So include this in your appendix and just your value in the results. And then, so this week you need to cite, obviously you're referring to an article, so you need to cite that in your text, and you also need to include the full reference in your reference section. And of course, this should be in the Ecology Letter Journal format. Don't forget to check for spelling, grammar, presentation, all that stuff, and your report is worth 7%. All right, and that is it for me. Thank you for watching.